The first thing I want to do is welcome you to the BERD Bootcamp series. We have an excellent speaker today. It is Dr. Michael LaMonte, and we are going to be starting in a moment. But before we do, I want to at least tell you that I want to just do a very, very quick introduction for Dr. Katia Noyes, who cannot be here at this time. She is away and she would love to be here, but it just didn't work out. So I'm going to do a quick presentation. I believe everyone can see my screen. It's the Core Competency Workshop series. It's the BERD mini series, Bootcamp. That's the first thing that we're going to be doing. And just to give you a heads up, we have a lot of Core Competency Workshop series, and there are some connections that can be made. Now, what you do need to know is the Core Competency Workshop series can be found in the Clinical and Translational Science Institute site. And if you look under Core Competency Workshop series, which is under Workforce Development, you will see a list of the ones that we have planned for this semester. Biomedical informatics, we've already done an introduction to, and there is a bootcamp video series you can watch. And obviously we're doing the BERD Bootcamp, and translational teamwork is coming up as well in November. Now, what's also very important for you to know is that we do have video recordings of most of our workshops, and that can really help you to kind of extend your learning. If you want to watch the video again, it will be there eventually. We just have to get it really worked through, and Liz is doing a great job of making sure the closed captioning is done. And what you also need to know is that you'll find other videos within the series that have been done in the past that might extend your learning with a quick connection to this particular workshop. These are all the workshops that we do have videos recorded for, and we actually looked through them to see which matches there might be. So we found that there are some matches in biomedical informatics and good clinical practice and health inequities and a few more. So if you do want to extend your learning, you can actually look at some of these other videos within the series and see if you can actually extend what we're working on today. What you do need to know is that today's workshop is study design and hypothesis formulation. But if you'd like to receive a certificate of completion for the workshop series, you have to attend the 4 workshops. And next week is developing a statistical analysis plan, followed by sample size and power, and reading and critiquing the scientific literature, which are 3 consecutive Tuesdays after this. You also need to know that if you do need this or really want to receive this, that there is continuing education credits, medical education credits as well. In order to receive this, you do have to attend all 4 within the series, and qualifying participants will be able to receive the 6 CME credits for the workshop series. So that is available if you do need it. I also want to tell you that halfway through the workshop, I'm going to be putting in the chat probably half an hour before the end, I should say, the BERD Workshop Series survey. It's really important that these evaluations are done. It is incredibly short. Basically, it helps us to report out to the grant in which we have in order to fund this series. And it also allows for the evaluations to improve the workshop series as time goes on. It's an incredibly short survey, and we ask that you complete it. I'm going to put the URL within the chat box, and then you'll also be sent reminders about it, just so that we do get surveys to it in order to inform basically the work we're going ahead with. So again, we want to thank you. I want to say that Dr. Noyes would love to have been here, but I'm afraid she wasn't able to do so. My partner who works with me, Elizabeth Bangert, is also on, and I am Catherine Sedota, and I'm the Workforce Education Specialist. Now I'm going to stop sharing, and I do want to tell you at least the formats. So Dr. Lamonte's going to be presenting, and if you have any questions, feel free to put them in the chat. He's going to stop about halfway through in order for us to take a break, or at least ask questions at that time. And at least 10 minutes prior to the end of the workshop, you're also going to have the opportunity to ask questions. So just to move forward, Dr. LaMonte, the floor is yours. Thank you.
Well, thanks again for that introduction. And also for those of you that are attending, thank you for joining the workshop. I want to make a point though. If you have previously attended the workshop or boot camp on this topic, you're going to see essentially the same thing. So if you've already heard me talk about this, I'm giving at least 95, if not greater percent, the same material as before. I just wanted to point that out. As we already heard, the BERD, Biostatistics, Epidemiology, Research Design, is part of the CTSI here at Buffalo. The BERD is a service-oriented component of the core in the CTSI. We do consultation. We offer collaboration when it makes sense, as well as doing some of the educational and training opportunities. And if you're interested more in what else the BERD does, as was mentioned, you can find us on the CTSI website. And then there's a whole menu of options that BERD offers. Already mentioned, but I'll just reiterate that these are the fall workshops that we're giving this semester out of the BERD. So I'll talk about designing studies in a very broad sense, by the way, and basics of hypothesis formulation. Next time, Jeff Mishinkowski, who's the chair of the Department of Biostatistics here at UB, he's going to talk about developing an analysis plan around a given study hypothesis. Greg Wilding, who's a biostatistician and director of the BERD, is going to talk about how do you estimate sample size and statistical power for a variety of scenarios. And then I'll be back as the final workshop. We'll talk about reading scientific literature in the biomedical space, how to critique it, and how to build upon it in our own research. So this is the objectives I'd like to put forward for tonight. Very informal, other than let's try, I'd prefer not to take questions during the talk, but I'll have a break in the middle. We can entertain questions about the first part of the talk then, as well as at the very end. I assume that you have little, if you will, or perhaps for some, no experience developing studies. This book that I've noted at the bottom is a pretty good 500-foot view of just about all aspects, from idea to dissemination in clinical research. It's not a bad book. You can get it as a PDF or buy a hard copy. It's not a bad book to have on your shelf or to share with students in the broad sense of covering what we have in this workshop series for you through BERD. So I want to start by just defining my perspective of science, and you'll see that this perspective kind of threads through some of the concepts I want to cover. Science, I think, historically has been thought of as a systematic process, so a methodical pursuit of unbiased knowledge in the face of uncertainty. There's a lot of parts to that definition. Systematic, I think, a very methodical, almost recipe-driven process. Unbiased means we want to know knowledge that is error-free. That's a tall glass of water, of course, to fill in the setting of scientific research. And then in the face of uncertainty, in that we can never be 100% sure we have the true answer. And as we'll talk about a little bit today, and as the biostatisticians that are part of this workshop series will emphasize, we have to declare, or we typically declare, how much uncertainty we're willing to live with. And that is what we call the alpha level of a hypothesis test. And I'll talk a little bit more about that, and so will Jeff and Greg. Very important from Mike's perspective to be concrete in knowing that we don't know everything. And as I just said, there is a level of uncertainty that, especially in the context of testing a statistical hypothesis, we have to declare, so that as we make a decision on whether we're going to reject or fail to reject the null hypothesis in the context of testing study effects, that we have some understanding of a gray area where we acknowledge there's uncertainty. Science is not a process of proving. So when you hear on the NBC News, Lester Holt says scientists at Harvard have proven that eating tree nuts reduce the risk for prostate cancer in men. No, that's not true, because the process of science doesn't prove. In fact, the process of science is a process of refutation, disproving or rejecting all possible challenges to our hypothesis. And that approach to science was made popular by Karl Popper, and it's referred to as falsification theory. Some of you may be familiar with that. So the idea of a scientific method is not new. This is a paper that I've often used in class settings, but published long ago in the journal Science in 1891, talking about the art of the scientific method, this methodical approach to problem solving. Another letter that was published in Science a little bit more contemporarily, 1986, by a fellow named Burke, makes an important statement that while we learn the disciplinary aspects of science, whether that's biochemistry, neurology, exercise science, nutrition science, that all folks who engage in scientific need to understand the philosophy of science beyond their degree itself. Why? Because that should unite us under some common umbrella or method, the scientific method to be precise, which in turn hopefully makes us better scientists as a whole or a collective. So this is just one way of looking at the scientific method. This comes from both the Burke article that I just referred to and a textbook on research methods. And basically, the process is what I want to show in terms of an ordered, methodical approach. First, coming up with the problem or the question. Generally, we come up with things that are way too broad to test in a feasible manner and form hypotheses around. So we typically have to clarify the problem. From there, we form a hypothesis, but it's got to be a hypothesis that's testable. We'll talk a little bit more about that here in a few minutes. Once we have the hypothesis, now we have to design a plan or design the study. Far too often, people come and say, I want to do this study. And one of the questions we'll ask them in the BERD consultation is, well, what hypothesis are you testing? And then you get the deer in the headlight look. Well, I don't know. Well, how can you design a study if you don't first have a hypothesis that you want to form a plan around in order to collect the appropriate information and then test the hypothesis? Part of that plan will involve, drawing off a study sample. It could be an animal model, could be a human model, but there's got to be a way to identify whom the study will be done on. Collecting the data, basically doing measurements of some sort on the study sample, using standardized methodology, again, an important part of the method itself, standardization. Once we have that, analyzing the data around the hypothesis test, at least as a primary part of the data analysis. And Jeff will talk more about that the next meeting. Once we have our data, trying to draw conclusions and understand how well those conclusions reach beyond the study sample to other populations, disseminating the findings through a peer review process. Peer review is a critical part of the scientific method. It's supposed to be a self-correcting part, even though when our paper gets rejected at the journal or our grants get trashed at study section, we often question how valuable peer review is. It should self-correct and ebb and flow so that on average, we have an unbiased approach to getting findings out and reviewing proposals for new studies. And then it starts over again. So this is really, again, just one example of how you might describe the method. The last thing I want to say about this, and I hope this is just reminding us all of this important aspect of science. Any project or study we do, form a hypothesis, go through the part where we have data, analyze it, and disseminate it. Part of that dissemination is that somebody else should be able to replicate what we did and see if they can confirm or if they refute our conclusions. And having this methodical approach, the scientific method, as a general framework is really meant to ensure that process of reproducibility. So what are some of the objectives that we're interested in biomedical and public health science research? Well, we're interested often in understanding diseases. So we want to know what disease is of interest. Sometimes certain disciplines refer to the disease or the what as the outcome. Some disciplines refer to the what as the dependent variable. We want to know who's affected. Is there any temporal or timing relationship to the disease and who gets the disease or who doesn't get the disease? Is there any geographical variation in the disease? Why does the disease occur? Often that why translates into us identifying risk factors or factors that predispose individuals to get the disease or to be protected against the disease. In some disciplines, these are called exposures. In other disciplines, these are the independent variables. Ultimately, in biomedical and public health research, we really want to get at the how. We want to know what the mechanism is that links the risk factor to the disease. Because if we understand a mechanism, whether it's fully explicated or it's partially explicated, we then have an opportunity for intervention. Whether the intervention is for primary prevention or secondary prevention or some other form of manipulating the host and changing the response given a certain susceptibility profile. And we want to do all of this, understand the what, who, when, where, why, and how as objectively as possible with a high, that means we don't skew the results to our own beliefs, as accurately as possible, accurately meaning as free of error as possible, and with reproducibility. Somebody else should be able to come behind us and do essentially the same study and then refute or confirm our results. Now, I want to refer to this sketch down here, this little schematic in the lower right, as a framework for what I'm going to talk about in some of the slides forthcoming here. Ultimately, we want to have information that is unbiased, that we can apply to a broader population than we're actually able to study. We may use an animal model to start with. We may eventually move to a human model. We may have a broader population in the entirety of the US or globally, but ultimately we can't measure entire states or the entire United States or entire continents. It's just not feasible. So 9 times out of 10 or 99 times out of 100, we are going to draw off a study sample. So we have to have a sampling plan. I'm going to talk about that momentarily. However, what's important to realize is that's the first step in our study process that gives us the opportunity for error. If we make mistakes in the sampling plan or executing a well-formed sampling plan, we can introduce error before we even take a single measurement. Once we have the study sample drawn off, and let's assume for the moment we had perfection with the sampling plan and executing it, then we have the smaller group of either animals or humans, let's say for purposes here, that we're actually going to do our study on. So we're going to measure the smaller group. That's the second major place where error can be introduced into our study and potentially threaten the ability to get unbiased knowledge or error-free knowledge. Measurement. And then lastly, once we have measures, we have data in place, we've analyzed it, what we need to understand is how well does the data we collected, the measurements we took on the smaller group, reflect back upon the larger group that we ultimately want to have the results reflect or apply to. And we use inferential statistical methods for that, and that's a third place where error can be introduced. So while the scientific method or science in general is the pursuit of unbiased knowledge, unbiased meaning error-free, if you look at just a very simple schematic of a study design and an execution of a study, there are all sorts of places in that where error can be introduced. And so being mindful of error is one of the most important attributes, I think, that the researcher, regardless of what model one's using, needs to be attentive to. Even if error is present, if you understand what its sources are and you have a way of understanding the likelihood that the measures are erroneous to some level, there's always ways to attempt to correct for that analytically. So understanding error is very important. Study designs, and now this is particularly towards human studies, can flow across different types of general study designs, observational studies, where we tend to watch people in the natural environment, and experimental studies, where we manipulate the participants, typically by assigning an intervention of some sort to one group and a control condition to the other. What I want to be very clear on is, while we often look at the clinical trial, whether it has randomization or not, the clinical trial is providing the highest level of information. The other study designs prior to getting to the clinical trial level offer us important insights that tend to build from one design to the next. In fact, I tend to have students think about study designs from the simple case study, what somebody observed in practice settings, let's say, a single individual, all the way down to a pragmatic community trial. That basically gives us a hierarchy of strength of evidence to support cause and effect. So study designs are very important to know weaknesses and strengths all the way across this hierarchy. One really good reason is, generally, we don't jump into clinical trials, in humans at least, without having a sufficient evidence base of observational data to warrant arguing the feasibility and ethics of a clinical trial are now timely. So a hierarchy of evidence, and that evidence builds across different study designs of different quality and strengths and limitations, ultimately pushing us towards making the decision, is it now time to do a randomized controlled clinical outcomes trial in humans? So which design do you use then? The choice of design can be answered by several different considerations. A really important one is the question being asked. Not all questions are amenable to each of the study designs. If intervention or manipulation of a condition to be an active condition versus a control or placebo condition is required, that almost pushes you towards the clinical trial design. If understanding a rare disease phenomenon, something that doesn't occur very often, let's say pancreatic cancer in a population, and what factors predispose to it, a case control study is probably the most efficient, because you go into the population, you select out the cases that are available, and you compare them to a group of individuals without the disease. And you don't have to wait long periods of time, for example, to accrue enough pancreatic cases to do the study. Resources is another big, and timing and logistics. All of these together may exclude certain designs just because they're not feasible. Randomized trials take a lot of resource, a lot of time, and they usually include complicated logistics. So that may wipe out the randomized trial design at a certain point of the evolution of knowledge and evidence. Other designs were, for example, the prospective cohort design, an observational study where you watch people over time for the development of new disease. If the disease takes a long time to develop, again, you may wait a long period before having enough disease cases. And because of that time constraint, that may not be the best design. But I think between the research question and considering some of these other things is the other really important consideration. What previous studies have been done? If there's a bunch of evidence published from a certain design in the literature, does it make sense to do yet another one of those designs? Or could you build an argument it's time to go to the next level of design? Also, what gaps in scientific knowledge or evidence need to be addressed? Sometimes the gap that's identified requires a very particular type of design to address that knowledge gap. And then lastly, and kind of going along with knowledge gap, the idea of equipoise. I don't know if this is a term that's familiar or not to you, but basically equipoise, or the idea of clinical equipoise as it's often applied to clinical trial design, is the idea that as researchers, it's our ethical duty when studying humans that the scientific process we're going to apply, the design, the question being asked, the measurements, the manipulations, that those are sound and that they're clinically relevant. That is, the result of the study has an ability to change clinical practice or public health recommendations, while constrained within that the highest level of ensuring the rights and safety of the participant themselves. That's clinical equipoise. So our job is to make the case that this question needs to be addressed by this design, let's say, for example, a clinical trial. And the reason is the results from this trial could provide new evidence that would change clinical practice and or public health recommendations, and that we can do it while preserving the rights and the ethical protection of human study participants. That's equipoise. So this schematic looks busy. I want to walk through it, and then this will be one of the last slides we come back to and see how everything else we talked about today fits into this. So anytime we sit down to design a study, usually that begins, or that process begins with a problem. We have a problem or a question, if you will, we want to address. Generally, from that point, we develop what's called the research hypothesis. Essentially, this is what we believe the answer is to the question or the problem. However, this is not what we test with the study. We do not conduct a study to see if our research hypothesis is true or false. We operationalize the research hypothesis into a testable hypothesis. Remember, I said the hypothesis has to be able to be made into or transformed into a testable hypothesis, and that's what we call the statistical hypothesis. This is what we actually observe and actually test as part of our study. The statistical hypothesis comes in two formats: the null hypothesis and the alternative. Our statistical tests that we ultimately use to make a decision on whether or not we can reject or fail to reject focus on the null hypothesis explicitly. It doesn't mean if we fail to reject the null, the alternative has to be true. That's not at all what that means. And we'll talk more about the idea of hypothesis testing and the mechanics of doing it next time. I think it's October 17th, when Jeff is with us, or with you all, talking about different types of statistical analyses. Planting the seed, though. The statistical bells and whistles that we throw at data are really there specifically to deal with the null hypothesis. So once we have a problem, we have a research hypothesis, we formulate that into the testable statistical hypotheses. Now we know how to design our study. We know the hypothesis we want to test. Now we can come up with a plan to sample humans. Most of what I'm talking about today, while it may apply both to animal models and humans, so I don't keep saying humans. Just assume I'm talking about human research unless I say otherwise. So we come up with a sampling plan that allows us sufficient study sample to do the hypothesis test. We have to have measurements to do the statistical hypothesis tests on, so we have to come up with a measurement plan. And then, as Jeff will talk about next time, we have to have an appropriate data analysis plan. Once we have that in place, we can proceed with the first part of the inferential process, and that is statistical inference. This is nothing more than a mathematical decision-making process based on probability regarding how compatible your observations are, your measurements are with the null hypothesis. Okay, so this first line, P, and then in parentheses, O bar HO, means what's the probability of making an observation or having a measurement, a mean value, let's say, in your study, if or given the null hypothesis was true. We can test that probability statement, and we do that with various statistical tests. Many of these I'm sure you're familiar with, from a chi-square to a correlation to a z-test or a t-test or an f-test. Those tests will ultimately, as I'll show you momentarily, yield the infamous p-value. The p-value gives us basically this p, the probability of observing your finding, your mean value, let's say, if you were doing a test of mean differences between two groups, the probability of observing your mean difference if or given the null hypothesis was true. There's other ways we can get around doing this. Sometimes we use what's called a confidence interval. I'm not going to speak much about that today. What I want to end my comments on this box called statistical inference. This statistical inference step is going to be only one part. This is very important that you realize this, and this is sometimes where there's a division of perspective. The p-value, the statistical test, and the conclusion we draw from it is only one part of understanding whether or not the original research hypothesis holds water. What else would we use, you may say? Don't we live and die by p-values? No, we don't. P-values is a piece of information. The broader scientific inference, which does in fact rely on statistical information, that which we get from our study, as well as that's already published in the literature, again, provides us one part or one piece of information. We also need to think about the existing literature in terms of the quality of studies that have been done relative to ours. Is ours better quality? Maybe carries a little more weight because we've addressed many of the flaws previous studies couldn't. The quantity of the evidence from previous studies, and how strong is it? Remember, strength of the evidence, study designs. Where are we getting the majority of the evidence? Cross-sectional studies, prospective cohort studies, randomized trials. And then finally, and I can't emphasize this enough, common sense has to be part of the scientific inference process. P-value does not usurp your brain or my brain. So does the finding make any sense from a logic perspective? Logic against what? Well, every discipline has an existing paradigm within which we make predictions, and those predictions should align with the overarching paradigm for what the question is. If you've ever read Stephen Kuhn's book, History of Scientific Revolution, it's the idea that paradigms from time to time fail, and the scientific process needs to reset itself so that we get back on track with having a paradigm within which predictions are made and answers align with what we think the truth is. So all of this is the evidence that should be used to go back and say, okay, was my idea more in the right or more in the not right space of thinking? Knowing that we live with uncertainty. We will never know 100% right or not right. Okay. So when we think about designing the study part, the protocol for a study often follows something similar to this layout. Granted, we could come up with a different table that has fewer things or adds more things. This is a general table. It comes after one that's in the Hulley book that I mentioned at the beginning of the workshop today, and that I said would be a good book to have as a general reference. So we have a question. That's part of the protocol. Okay, why do we need a study question? Because that study question should focus the aims or the what are we going to study. Most of the time, the questions are too broad and they need to be narrowed down so that they're manageable, so that you can make testable hypotheses, and so that the what takes on meaning at the end of the day when you have your study completed. The background and the significance part of a protocol. This is what should support the rationale for the study. In other words, the why. Why is it important to do this study? Think about a scientific manuscript. The introduction to a manuscript is essentially the background and the significance. It should build the argument of what's known, what's not known, and most introductions end with something along the lines of, therefore, the objective of this or the purpose of this study is. So why do we need to do the study? And that gets stated fairly clearly at some point. This could be a grant as well. Background and significance leads to the specific aims of the grant. The why do the study and the what then should be, at least in part, directing the hypotheses that we're going to form, and particularly the testable hypotheses. So the aims themselves, the question as it's further refined into what we're going to study, ultimately should direct development of the hypotheses that are going to be tested. There can be a single hypothesis, there can be multiple hypotheses. We'll talk a little bit more about that momentarily. At that point, you have the question, you've justified the question, you've even explicated your hypothesis. Now it's time to design a study to allow this to be done. Feasibly and as unbiased as possible, test the hypothesis. So you have to pick a design. And this is the how will the study be conducted part. Is it going to be observational, experimental, and then the nuances within either of those broad categories? Part of the design is going to be identifying the participants. This participant pool is ultimately going to lead you to the study sample, and that's within whom you're going to take measurements for hypothesis testing. So who will be studied? Typically, we do this employing a couple of pieces. There could be more. I'm trying to keep it kind of on the simpler side. But in many studies, there's inclusions and exclusions. That helps us to narrow the scope and in some instances control for potential biases. So we exclude them. Sampling design. How are we going to get the people from the broad into the narrow study sample? How are we going to go out, recruit them, and make a decision on who gets sampled into the study population or the study sample? All of that is part of the participant element of the protocol. Measurement is obviously a big part of a study design. If we know the hypothesis we're going to test, there should be an independent variable and a dependent variable, at least as part of the hypothesis. We have to have measurements that allow us to get accurate, reproducible, and feasible measures of those traits so that we can employ hypothesis testing around the quantitative or the qualitative aspect of those traits. Okay. Again, some disciplines refer to the predictor variable as an exposure, or the independent variable, or what you're manipulating if it's a experimental design. The outcome is equivalent to the disease, or if it's a disease you're studying, or the dependent variable. Some disciplines will refer to that. And then there's often other relevant information we want to collect. And in many disciplines, we call that information covariance. So measurement, each one of these, the predictor, the outcome, and the other stuff needs to be thought of in terms of a measurement protocol that optimizes collecting what you think you're collecting, internal validity, and minimizes error, whether it's random error or systematic error. I'll talk a little bit about those momentarily. And then finally, once you have a measurement plan, again, the measurement plan should be supporting getting you what you need to do a hypothesis test. Then you design the statistical aspect of the study, which Jeff will talk about on the 17th, which includes how many people do I need? How many people do I need in order to test the hypothesis? And part of that, how many people do I need, aligns with another statistical concept called power, which I'll talk about in a moment. And Greg Wilding will give you some examples of how to calculate power in the third session of this workshop. So sample size and power, again, these are directly related to the test, the hypothesis test. And then the analytic approach or the data analysis technique, that will also depend on the design of the study, the sample size, and the hypotheses being tested. So a lot of this stuff interconnects. We can lay it out like this, but really, once we start right up here, what's the question? So many of the what question do we want to answer will get us down into some of these other levels even before we formally say, okay, we've nailed this. Now let's move to the next. Now let's move to the next, and so on. Because so much of this interrelates up and down, if you will, in this table. So the question is often a generalization. As I said, most research problems in question are too general. They need to be narrow. So for example, one question we may ask is, should people eat fish? Is it good for the health, or is it bad for health? We can narrow that question as such. What is the frequency and how much fish is eaten by US citizens or US adults? And we could define adults as greater than or equal to 20 years. We could ask, are there adverse effects, such as mercury toxicity, with fish eating in adults? That's a specific question. How much and what type of fish need to be consumed to reduce the risk of a certain disease? That's a very specific question. And then also, do fish oil supplements have the same biologic effect on disease as dietary fish intake? Should people eat fish is a broad question under which each of these four much more specific questions fall. And we could design a study around each of these explicit questions far easier than we could if we said we just want to tackle, should we eat fish? In fact, we may find that's just too broad. And we would say we need more specificity. And we'd find ourselves down at one of the four or any other number of possibilities to refine and narrow that question. So carrying forward with that example of fish consumption, I want to point out that across study designs, and as I said, these designs build a hierarchy of quality of evidence towards cause and effect. We can ask the exact same question and move upward in the chain of quality of evidence as such. Cross-sectionally, a cross-sectional study is a study where a group of people are measured at a single point in time, and we simultaneously collect information on the independent and the dependent variable. So the big thing that's lacking here is temporality. You don't know which came first. Did somebody who happened to have heart disease at the time you did your cross-sectional study also happen to eat more fish because the heart disease got them to the healthcare provider? The healthcare provider said, eat more fish, it'll help you in the long run. Did somebody who happened to eat more fish naturally have less opportunity for heart disease to fall into your cross-sectional study as a heart disease case? We wouldn't be able to tease that apart. But what we could do as a starting point is answer this question. what's the relationship between existing heart disease with the type, frequency, and amount of fish consumed by adults at a certain point in time? And you may say, well, Mike, where would we get this information? Well, you could build a study to collect it. We could survey the University at Buffalo tomorrow, have people fill out a questionnaire and ask them about fish consumption and whether or not they've ever been told they have heart disease. And there we have a cross-sectional study we could pursue. Or we could use existing data resources, like the National Health and Nutrition Examination Study, or NHANES, where these sorts of cross-sectional studies are published frequently. Once we had an understanding, and let's say what we found was people who had a higher intake of fish had a lower prevalence or likelihood of being a heart disease case at a single point in time. Let's say that was our finding. We could take it to the next level and say, well, we want to start trying to figure out which came first. The next design we might go to is a case control design. Now, in this design, we select two groups of people. We don't just take one comparison group. The idea here is, right, our hypothesis here is that the reason the cases ultimately got sick and became cases is because they had something in the past that predisposed them to become a case. And for the controls, or the comparison group without the disease, there's something about their past on that factor that was absent or less frequent. And so the question here might be, is history, past, fish consumption different between those with heart disease and those without? Okay. The difference between these two designs is, in the case control, we're trying to figure out the past, assuming that the order of case occurrence and control occurrence is being driven by something that occurred in the past. From there, we could go to a better design that starts to approximate timing, and that would be the prospective cohort design, where we identify a group of people who are without the disease. They don't have heart disease. And at the beginning of the study, we assess their fish intake, maybe by a questionnaire. And then over a period of time, usually it's several years, 3 years, 5 years, 10 years, we identify people who newly develop the disease of interest. In this case, it would be heart disease. And then what we evaluate is, did their fish consumption from none to some, let's say, or none to any, or you could say none, a little bit, and a lot, however you want to define it, did their fish consumption, while they were still free of disease, predict who got disease and who didn't? Now we have the timing. We know that the disease came after the independent variable or the exposure of interest. But prospective cohort studies, while they give us some better assessment of timing, there are some things that occur in populations of individuals that we cannot always account for, and that can introduce error or bias into the results. So if we had enough evidence from cross-sectional, case control, and cohort studies that at least was consistently suggesting more fish consumption was related with less heart disease, we might be able to make the argument for equipoise and propose a trial now needs to be done, or an experimental design, to get a definitive answer. And what would be that design? We would take two groups of people. Again, these people would not have cardiovascular disease, so that part's very similar to the prospective cohort study. But instead of just giving them a survey and taking fish consumption as it falls, we would assign one group in the randomized trial to be exposed to high fish intake, and another group to be a comparison where they were the control condition, either none or little fish intake. And in fact, today, one approach that's used so you get out of the issue of dietary intake and all of the other things that come with how it's prepared, where it's caught from, and so on, is to use a fish oil supplement or an omega-3 pill. So we might design a trial where one group gets randomized to omega-3 fatty acid supplementation and the other to a control condition. And then we follow them and we see who gets heart disease and who doesn't. The randomization, as we'll talk about, because getting into either group, the intervention with the fish oil or the control group for the comparison arm, it's done by probability. So each person has a 50% probability of going into one group or the other. So at the end of the day, when you have both groups enumerated, it should be balanced across the groups on everything related to the individuals, both measured and unmeasured, including genotype. So that's where the power of randomization comes into play. However, the argument to do a randomized trial where we're actually manipulating humans to an intervention that could either provide benefit or cause harm, the evidence building towards that has to be substantial so that we can maintain clinical equipoise in the ethics of doing human subjects manipulation. So what is it about the question then that we want to think about when we're writing it out, literally, writing or typing it out? And one little moniker is finer. The question should lead to a feasible study. It should be interesting, novel, ethical, and relevant. Now, I've pointed out a few things here. So feasible, this is a big ball of wax. So if a study requires a large number of participants, big sample size, in order to have adequate power, that may be deemed less feasible. If a study, particularly a clinical trial, is going to cost a lot of money and a lot of time, that may be deemed as infeasible, regardless of the evidence suggesting that is the design that should be done next. Interesting. The question has to be intriguing to the general scientific community. Why? Because they're the people that are going to review your grant. Okay. So there has to be a general intrigue in the scientific literature or the scientific community, at least within a given discipline. Novel, and I point out here in blue, innovative. This, as well as relevant or significant, are two of the main NIH grant review criteria. And these are sometimes two of the hardest elements of the research question to nail down. Novelty maybe isn't so hard to know whether your question's novel or not, but what comes up to be an issue is, does it have to be novel? Can a question that's going to add additional knowledge in an area still be something that's relevant, even if it isn't novel? So what is novel? That's something that often plagues us in reviewing grants or even sometimes reviewing manuscripts through peer review at scientific journals, if the journal requires novelty to meet the small percentage of papers they ultimately publish. So is it a cutting edge method? Is it something where you put on the table a controversy and you say, we're going to try and address it here, and we can address it because? Okay. Relevance or significance, this is a really important one too in terms of scientific review at NIH and also peer review at the level of a journal, and it's a little bit more subjective. There's no textbook of relevance that I'm aware of in a library or textbook of significance when you become a grant reviewer and they give it to you and it's 1,000 pages and you have every possible iteration of what is significant. So it requires a little bit of common sense, understanding of the scientific knowledge within a certain domain or question area. I think at NIH, they're really interested in, does a study have the potential to change clinical practice or at least inform clinical practice or public health recommendation? And is it going to be relevant to the next generation of studies or research that will follow from the findings from the particular question you're trying to develop? I think any of these are things that need to be considered, and all of us get better at finer if we master the literature. You've got to know your content area. I don't claim to know things outside of the area I work in, but I will be bold to say I feel like I'm a very strong consumer of the literature in my little niche that most of my research occurs in. Being skeptical is very important. And that means we have to be skeptical about our own ideas or our peers' ideas, our colleagues, our students. Sometimes a little bit of tough love at the front end saves a lot of bit of strife through a process or at the back end. Creative and tenacious, we all have to have thick skin. We've got to keep trying to come up with good ideas, try and vet them through some framework like Finer or some other one that you'd prefer. But then I think one that we don't talk enough about, especially as our careers develop and we go from early career to mid-career to later career, even later career investigators need mentoring. Finding a good mentor who is willing to tell you what they think based on experience, even if it's at the cost of a few tears, and someone who we're willing to go to because we know they'll be honest with us. I don't think that piece of developing research that is using a mentor that you trust as a foil is something we give enough credit to, and it's probably not something we get trained well in as we move through our formal training processes and our post-formal training processes as mid and later career investigators. So Finer, or some analogous scale to think your ideas through. Now, hypotheses that we flesh out of the research question may actually follow over multiple layers. So every study should really have a very clear primary question, or it may be the sole question. But if there's more than one question being addressed, there should be something defined as the primary question or aim. And that has a primary hypothesis. So a statistical plan, a sampling plan, a measurement plan that's going to all feed into having this primary hypothesis test be possible. There may be additional opportunity because of the data being collected to look at secondary questions. Some secondary questions and secondary aims you may actually further develop into secondary testable hypotheses. Some secondary questions are more exploratory. Exploratory questions, whether they're secondary, tertiary, quaternary, or however many they end up being down the list, these may be really what they are. We're looking at data almost as preliminary data, pilot studies within a bigger study where data are available. And so they may not be suitable for testing hypotheses. They may actually be more amenable to develop new hypotheses that can be addressed better in a different study setting. And in terms of translational research, which is what the CTSI here at Buffalo and the BERD kind of helps to think us all through, it's really an intersection of three major domains: laboratory research, clinical practice, and population-based research. And so the translational aspect, as we cut across this continuum, which is really actually, I think, circuitous, it feeds forward and back on itself constantly. There's a translational domain within there where we do have to think about how are we informing that next level of research, or informing back on the previous level to enhance the research process as well as the outputs, patient care and public health. So specifying a study hypothesis is probably the next most important thing, in Mike's opinion, to the development of a strong question or problem. We really want to have a very clear study hypothesis, testable hypothesis, so that the rest of the plan can be appropriately built around that. This focuses the question, and in doing that, it gives us an understanding of who we need in the sample, what the variables that we need to measure or collect are, and it really guides development of the analysis plan. what does the statistician need to think of, or whomever is going to do the analysis? We tend not to use hypotheses as much in descriptive studies, like a cross-sectional study, where we may be generating an idea that then allows us to build a testable hypothesis for a better design study. Mostly analytic studies, such as cohort studies and clinical trials, where we're looking at evaluating differences and we have some idea of timing of when the independent variable and when the response or dependent variable takes place. And then the other thing the hypothesis is critical for, and the more explicit it can be, the more it can inform this critical last bullet, and it will help us understand how large the study has to be, the sample size, and equally, if not more important, what statistical power do we have to test the primary or however many hypotheses there are, tightly related to the hypothesis and how it's written, as Jeff will talk about and Greg will talk about as well. So I want to talk a little bit more about the hypotheses now from a testing standpoint. And again, Jeff and Greg will give you more detail from the statistician's perspective. This is more from a conceptual design standpoint. As I've already said, the null hypothesis is the big player. This is what we subject to the statistical test, the statistician's role in the study often. And it's basically a statement of no difference or no relationship between study groups. There'll be no difference in mean body weight between group A and group B, whether it's observational or whether group A and group B represent an intervention group and a control group. The null says there's no difference between the two groups on what we're measuring. Assuming that the truth is no difference, that is, the true answer to the question in the universe is that there is no difference, so that assuming the null is correct, statistical hypothesis testing estimates the probability of finding your mean difference or your association if, in fact, the null is true. I'll show you what I mean in a minute. The alternative hypothesis is simply the opposite of the null. If the null is there is no difference between groups, the alternative is there is a difference between groups. Okay. The alternative is not tested directly. It's basically the reciprocal of the null. If you reject the null, then the alternative is what you're left with. But again, it's not directly tested as a mathematical function of statistical analysis. More or less, it's accepted if we reject the null. Some of you are probably familiar that hypotheses can be stated in more than one format, typically one or two-tailed or one or two-sided. This is what we mean by directionality. So when stating a one-sided hypothesis, you're not only saying that there is going to be a difference as an alternative, but you're going to say in what direction is there going to be a difference. So using the fish consumption in heart disease will be lower in consumers versus non-consumers. That's a one-sided hypothesis. That means we're developing our study to test the null of it won't be lower in consumers versus non-consumers. The opposite of that is, in the more general approach to hypothesis testing, is non-directional or two-sided, where all we're saying is that there will be a difference, but we're not specifying in what direction. Therefore, the null is that there'll be no difference. No pun intended, but what's the difference here, Mike? The difference is one-sided hypotheses generally have more or greater statistical power. However, if the direction that you specify to test is wrong, you may not have sufficient power to, statistically at least, identify the direction opposite to what you specify. So unless there's a really good reason Most of the time, we operate with a two-sided hypothesis test. While having slightly less power, we're not committing ourselves in one direction or the other. So we should have an ability to look at something that's higher or lower in one group compared to the other. There's one exception to this. The one exception is the randomized trial. By definition, it's a one-sided hypothesis test. The hypothesis being tested is that the intervention will lower risk or will benefit the intervention group as compared to the control condition. It wouldn't be ethical to test a two-sided hypothesis in a randomized trial, let's say, of a new drug. While we are looking at both the benefit as well as any side effect or adverse effects that may be identified, we are hypothesizing benefit. Otherwise, the trial is not going to go through IRB or get funded by NIH. That's an exception where really the hypothesis is always going to be one-sided or direction. So what do I mean by hypothesis testing? What's Jeff going to talk about with the analysis plan? What's Greg going to talk about with statistical power? This may be familiar, and if it is, let's see it as a refresher. If it's not familiar, I'm going to walk through this. Basically, as I've already mentioned, hypothesis testing is nothing more than evaluating how compatible your observations are, the mean difference, let's say, in your study, how compatible that is with the null hypothesis, remember, which is a statement that there will be no difference in the means between groups. So when we do a hypothesis test, we make the assumption that there's two sampling distributions for the observations, the means, let's say, or the probabilities or the proportions, whatever it is you're measuring. There's two distributions. There's a distribution under the null, where we find that a difference between two measurements equals 0. There is no difference. And then there's a distribution under the alternative, where we find that the difference between two groups is not equal to 0. Okay. When we do a hypothesis test, we do it essentially as a truth table. And there's a few, I'll show you the truth table in a minute, but there's 3 areas under these intersecting sampling distributions that you need to be aware of. And you've probably heard them, at least as a term, but maybe weren't quite as sure what they meant. First of all, there's alpha. And you may hear alpha was 0.05. Okay. Alpha is the area that's above a critical value, I'll tell you what that is in a minute, that belongs to the null hypothesis. Now, what's the critical value? A critical value is the test statistic, the size of the test statistic. If it's a chi-square, it's the size of the chi-square itself. If it's a t-test, it's the size of the t-value. If it's an f-test, it's the size of the f-value. It's the critical value for the given statistical test that the statistical result has to be greater than in order to reject the null hypothesis. So when we do a statistical test, we're looking to see if our statistical parameter, the f, the chi, the t, is larger than some pre-specified critical value on that test. Okay? So back to alpha. Alpha is the area that falls above the critical value. Remember, things that fall above the critical value should poise us to reject the null hypothesis that there's no difference, and then assume that there is a difference. However, alpha is above the critical value, but it still belongs to the null. So what we often refer to this is uncertainty. This is what I meant by alpha, the level of uncertainty in doing a statistical hypothesis test. There's going to be a small percentage of times we do the hypothesis test for the given data distributions we're working with, where we have a test statistic that actually is bigger than the critical value, but it represents the null value or the null distribution. And if we reject the null hypothesis, we're making a mistake. That's called an alpha error. Beta is another area that's important to know, at least conceptually. Beta is the area below the critical value. So the test statistic wouldn't achieve being greater than the critical value. So our decision would be to not reject the null. But beta is the area below the critical value where it actually belongs to the alternative, and we should be rejecting the null. And this is called a beta error. Okay. Lastly, statistical power. What does that mean? What are the statisticians doing when they tell us we have 80% power, we have 90% power? Power is 1 minus beta. So power is the area under the alternative distribution minus that small amount of area from the alternative that falls below the critical value that we would have mistakenly not decided to reject the null hypothesis with. So 1 minus beta, or your statistical power, is everything under the alternative, taking into account how much error you're willing to make in accidentally rejecting the null when the null shouldn't be rejected, the alpha error. Okay. I'm going to move to the next slide and bring the truth table in, as hopefully this reinforces what I was just saying. A statistical hypothesis test is nothing more than comparing a state of truth, which we assume is the null, there's no difference, against a decision based on our data and the statistical test, which is to reject the null or fail to reject the null. When we cross-tabulate our two possible decisions against truth, we have the ability to be right twice, and we have the ability to be wrong twice. The two errors that occur with hypothesis testing is the alpha or the type 1 error, and that is where we, right here, incorrectly reject a true null hypothesis. Why? Because the value of our statistical test fell above the critical value, but in terms of probability, that was within the boundary of alpha, and the data actually belonged to the null distribution as opposed to the alternative. That's the first error, type 1 error. The second error, or type 2, or beta error, is where we fail to reject a false null hypothesis. And why does that happen? Because our test statistic isn't big enough to exceed the critical value. So we assume our observations belong to the null distribution when in fact they're part of this little tail of the alternative distribution that we label as beta. Okay. 1 minus beta, right up here in the upper right, this is power. This is the ability of our data, when subjected to statistical hypothesis testing, to generate a critical value that is exceeded by the test statistic, and therefore our decision would be to reject the null, and we would be doing so correctly some percentage of the time, after accounting for how much percentage of the time we're willing to be wrong with the alpha value. So this is kind of the mechanics of the hypothesis test. When Jeff's with you next week and when Greg's with you the following, they're going to give you actual examples of a t-test, a chi-square test, an f-test, and put this into some tangible understanding with some examples using data and some published information. The last thing I want to say before we take a quick pause is be careful with the notion of multiple hypothesis testing. So the summary over the concept of hypothesis testing is it's either going to be one or two-sided, directional or non-directional. It's going to be based in part on the effect size. So how big of a difference do you think there's going to be in the mean value or the proportion between two groups? It's going to be dependent on the alpha level, which is preset, alpha 0.05, alpha 0.01, alpha 0.10. This is all something we're in control of. This notion of 0.05 being the magic number is malarkey. It's just a conventional number that has historically been used. But there's times where we want to have a much more conservative hypothesis test. Okay? 0.01 is more conservative than 0.05. An example being when we're doing clinical trial on an early experimental drug, we don't want to be wrong in saying we reject the null that it's good for people if it's really going to kill them. So we might test the hypothesis with a more stringent alpha level. Power I've already mentioned. The idea between hypothesis testing is to minimize both sources of error, beta and alpha. And that's done by increasing sample size and by having really small error in your measurements. And then lastly, I mentioned on this slide when I first turned to the slide, multiple hypothesis testing. The more times you run a hypothesis test on the same data set, the more likely you're going to reject the null by chance alone. So if you have a data set and you run 100 statistical tests on it, okay, there's a percentage of tests that'll turn up to be sufficient to reject the null, but they'll actually be alpha errors. And so if you plan on doing multiple hypothesis testing on the same data set, there are correction procedures to minimize the alpha error. The trade-off is it makes the critical value larger. So you have to have a bigger effect to surpass the critical value. But by surpassing a bigger effect as a critical value, you're enhancing your confidence that I'm not making an error simply because I'm running the data several times through the hypothesis testing mill. So from a statistical test standpoint, this is a generic form of a statistical test. So this is the test statistic for a z-test. And the reason I use this to illustrate what a statistical test is doing in this concept of hypothesis testing, it's based on two things. Most statistical tests are based on essentially the same two things. The difference or the effect size, in this case, it's how big of a difference is there between your observed mean and what you might assume is an expected mean. The bigger that difference is, the bigger the z will be, assuming the denominator stays the same. So one way to enhance the size of the statistical test that will allow you to be more likely to pass over that critical value is to have a big effect size. Sometimes you can't control the effect size. You don't know what it's going to be, or you can't manipulate the participants enough to ensure a big effect size. So if the effect size stays relatively constant, the other way to enhance the size of the test statistic is to minimize error. S here stands for the standard deviation. So a z statistic is nothing more than the difference between two means divided by the standard deviation. So any test statistic is going to be driven by these two phenomena. How big of an effect size are you modeling? And how much error is there in your measurement? How much variability is there around the actual mean value? Okay, the larger the standard deviation, the smaller the z at a constant effect size. If you have a lot of noise or measurement error, the only way to offset that is to have a bigger effect size in order to get a z-test, in this case, that should be sufficient to overcome the critical value and allow you to reject the null if it's truly false. Okay. Both of those, the effect size and the error or variance, is a critical part of study design. Planning for the effect size, having good measurement of what you're looking at for mean difference, and then having as little noise or measurement error as possible. That will make the Z as big as you're going to get it to be based on your study and give you the best chance during the hypothesis test to be larger than the critical value and thus reject a truly false null hypothesis. Let me just, we better stop there. Questions?
Great. Well, we definitely have one in the chat, and it is, what are areas in the process where you recommend seeking advice from a mentor?
Okay, so if I'm understanding the question, this is what I think you're asking me. Which part of the research design
I think that's what she means. That
should you look for?
Selena,
Frankly, I think,
yeah,
pardon me?
Selena can unmute and tell us if she wants anything different, but I think that's what she's saying.
Okay. My opinion, and that's all I can offer you is Mike's opinion based on my own experience, is that every stage, it wouldn't hurt to get somebody who's been there, done it a lot, and has been successful to give some feedback. Is it a good question? You know, one of your colleagues or somebody you know through someone you know may not be at your institution, but that person happens to sit on a study section at NIH or is a journal editor or associate editor who does a lot of reviews for journals. Is this a good question? Am I going to get movement on it? Or is this the right design? Or am I choosing the right statistical test? Or am I interpreting the data? And that's a good place for us all to have some fresh eyes and thought, particularly people who have kind of worked through some of the nuances by trial and error in bringing a study finding into the view of peer review and through the dissemination of various science outlets. Are we correctly interpreting the data? Are we placing too much emphasis on one place and not enough on another? Are we overreaching the conclusions? That's something we all often perhaps do from time to time, is we want our study to say more than it's actually capable of saying, or at least say more than the data can substantiate. So having a mentor help us calibrate that, I think, is invaluable.
Excellent. Thank you. Does anyone else have any questions? If not, I'm going to stop the share, the recording.